Welcome back. So this is the beginning of the third and final video describing the TCA cycle, all of the enzymatic reactions and the mechanisms associated with those reactions. So we're going to begin this time by talking about reaction 6, which is catalyzed by succinate dehydrogenase. So once again, this is a dehydrogenase, so you already know that this reaction is going to involve a hydride transfer. But this is a little different than the other reactions that we've looked at in the TCA cycle, because here you can see that in this reaction, succinate is oxidized in order to reduce FAD to FADH2. So succinate is oxidized to fumarate to reduce FAD to FADH2. So here, the hydride transfer, instead of being used to make NADH, is going to be used to make NADH2. So a couple of things to note about this reaction. So this is a stereospecific dehydrogenation. So like many of the reactions that we've seen before, there is only one stereospecific product, so only one stereoisomer. In addition, we already know that this reaction is inhibited by malonate. So I'd like to talk a little bit about why this reaction uses FAD as opposed to using NADH. So if you remember from Selim-Malek, FADH2 is slightly lower in energy than NADH. In fact, when it contributes its electrons to the electron transport chain, the electrons from FADH2 yield enough energy to make about 2 ATP per molecule, while the electrons from NADH yield enough energy to make approximately 3 molecules of ATP per molecule. So this is a less exergonic reaction, and this is actually really important here because here what's happening is we're going from an alkane to an alkene. So we're going from a single bond to a double bond that's present in fumarate. So that's our oxidation. So this doesn't yield enough energy to make a molecule of NADH. So in all of the reactions that we've seen before where we made NADH, this is oxidizing an alcohol to either an aldehyde or a ketone. So that's a more exergonic oxidation than what we see here. So because the oxidation doesn't yield as much energy, that's why this reaction has to make FADH2 as opposed to NAD. However, one very interesting thing about this reaction, this is the only enzyme in the TCA cycle that actually takes place in two processes. So succinate dehydrogenase not only plays a role in the TCA cycle, but it also plays a role in the electron transport chain. And because of this, succinate dehydrogenase is actually located in the mitochondrial membrane. So all of the other reactions that we've been talking about that are a part of the TCA cycle, all of those reactions take part in the mitochondrial matrix, which is inside the inner membrane of the mitochondria. In contrast, the succinate dehydrogenase enzyme is actually embedded within the mitochondrial inner membrane, just like those members of the electron transport chain, where it can easily pass on the electrons from FADH2. So that's something really different about this reaction. So a little bit more about how this reaction occurs. Why this is important that it's embedded within the electron transport chain is because this FAD molecule doesn't dissociate from the enzyme, unlike the NADHs that we've seen before. So here, FAD is actually covalently associated with a histidine residue that's present within succinate dehydrogenase. So it's covalently bound to this histidine in the active site, and it can't dissociate from the enzyme itself. Also making it very important that this enzyme is embedded within the inner mitochondrial matrix, where it can donate these electrons to the electron transport chain. So we're not going to talk about the mechanism of reaction 6 in the TCA cycle as a part of this video because we're actually going to focus somewhat on that mechanism and how FADH2 is reoxidized by coenzyme Q in the electron transport chain in the next chapter. So this is all we'll say about the mechanism of enzyme 6. So now let's move on to reaction 7. So reaction 7 is catalyzed by the enzyme fumarase, and fumarase takes fumarate, reacts it with a molecule of water to produce L-malate. So we're going from fumarate to L-malate. So this is essentially a hydration of a double bond. So basically we're adding a water molecule across this double bond. So here you can see we added a hydrogen and a hydroxyl group, and that's what leads to the production of malate. So if you remember, the elimination reaction and the opposite of the reaction, which would be a hydration of a double bond, this can go through either a carbocation or a carbanion intermediate, depending on whether you add a hydroxyl group or a hydrogen or a proton to the substrate first. So in this case, there are two possibilities. So the fumarase mechanism, so we're going from fumarate to malate. So in order to go through the carbanion, what would happen would we would first add that negative hydroxyl group, which would push the electrons from the double bond out onto the carbon, forming a carbanion. Then second, if we added the proton to that carbanion, now we have the product malate. The other possibility is if we first added the proton, this forces the electrons to come back to the electrons from the double bond, or what the proton is actually added to, leaving this carbon as a carbocation, which then can be resolved by adding the negative hydroxyl group. And once again, we have malate. So in this case, we actually know experimentally the mechanism of fumarase goes through this carbanion transition state. We know this because carbanion analogs are very good inhibitors of this enzyme, which indicates that the transition state must involve some kind of carbanion. So this data together indicates that fumarase is actually going through this mechanism where the hydroxyl group is added first to form the carbanion transition state, and then the proton is added to make malate. So the last and final reaction of the TCA cycle, reaction 8, is catalyzed by the enzyme malate dehydrogenase. So malate dehydrogenase catalyzes the oxidation of malate to oxaloacetate, which reduces a molecule of NAD to form NADH. So this is very similar to what you've already seen for isocitrate dehydrogenase. If you remember, when we talked about isocitrate dehydrogenase, we said that we removed a proton from the hydroxyl group and we removed a hydride from the carbon. The hydride is transferred to NAD, and when this happens, this forms a carbonyl as opposed to the alcohol. So this is a case where we're oxidizing an alcohol to a carbonyl in order to reduce NAD to NADH. So this reaction is actually highly unfavorable. So you can see here that the delta G is positive. It's actually positive approximately almost 30 kilojoules per mole. Because of that, this reaction actually leads to very low levels of oxaloacetate, meaning that this reaction is actually coupled to the next reaction in the cycle. It's coupled to the citrate synthase reaction. And so overall, the coupled reaction is favorable, but this wouldn't happen if it weren't coupled to the other reaction. So because we've already discussed the mechanism, I'm not going to discuss this mechanism any further. You can refer back to the isocitrate dehydrogenase mechanism. So that's the end of our reactions of the TCA cycle, but I just want to go back and summarize all of the products that are associated with the TCA cycle. So in general, first, pyruvate is converted to an acetyl group, to acetyl-CoA by the pyruvate dehydrogenase complex, and that acetyl-CoA group is what enters the TCA cycle. So in the actual cyclic part of the TCA cycle, one acetyl group is oxidized to two carbon dioxide molecules. So this leads to the transfer of 4 electron pairs. However, something that's really important here is that it's not the actual carbons of the acetyl group that are oxidized and released as CO2. So in fact, on Friday, we are going to do an exercise where we trace the carbons. So make sure that you're very familiar with all of these reactions and that you bring your notes with you to class on Friday, because we're going to use your notes and what you've learned in order to trace the carbons to the TCA cycle. So in total, the cyclic part of the TCA cycle yields 3 NADH, so 3 electron pairs were transferred, 1 molecule of FADH2, and 1 molecule of GTP, which can easily be converted to ATP per pyruvate. If we add that to the additional NADH that was produced by the pyruvate dehydrogenase complex, then per glucose, together the pyruvate dehydrogenase complex and the TCA cycle yield 8 NADH per glucose, 2 FADH2, and 2 molecules of GTP, which can be converted to ATP. So the NADH and FADH2 that are made from these processes, as well as from glycolysis, those electrons are going to be passed to the electron transport chain, where they're going to be used to reduce oxygen to water, and the energy from those electrons are going to be used to produce ATP. So it's approximately 3 ATP per NADPH and 2 ATP per FADH2. So together, the cyclic part of the TCA cycle actually generates approximately 12 ATP. If we add that to the NADH from the pyruvate dehydrogenase complex, that would be an additional 2 NADH. That would be approximately 18 ATP together. So as I mentioned, we're going to trace the carbons in the next class, so you should be ready for that. And also, surprise! Thank you for watching these videos and being a responsible student and doing your biochemistry while I was gone. So as a part of this, as the next class, I also want you to bring the following as a point in extra credit. So if you bring on Friday the answer to this question, what is your favorite TCA cycle enzyme and why, then you'll receive 1 bonus point. Now note, this has to be brought to class on Friday. You can't bring it late because that shows me that you didn't watch the video. So congratulations on your bonus point. So the last thing I want to talk about is the energetics that are associated with the TCA cycle. You can see here that there are 3 reactions that have a negative delta G. Most of the reactions under normal substrate and product levels are actually operating very close to equilibrium. So most of these reactions can go in either direction. However, the citrate synthase reaction, the isocitrate dehydrogenase reaction, and the alpha ketoglutarate dehydrogenase reaction are all operating at a negative delta G. So this should tell you that all of these steps are going to serve as control points for the TCA cycle. And in fact, that is the case. These 3 enzymes tend to respond to several different control mechanisms. First, they respond to substrate availability. So if there's lots of substrate available, then that tends to activate the enzyme. This makes sense because this would indicate that there was a backup in the TCA cycle, so we need to continue moving forward. They also respond to product inhibition. So if we've made too many of the downstream products, it's going to inhibit those enzymes. That also makes sense because that suggests that we're starting to build up more and more energy, and then these products are backing up because we don't need any more ATP. And if that happens, that's really important to know because then some of these products can be sent out from the TCA cycle. For example, can be converted into oxaloacetate and can then be used for gluconeogenesis, or some of these products can also be used for amino acid biosynthesis. And finally, they operate by competitive feedback. So in many cases, downstream products that are consistent with higher or lower energy can either activate or inhibit the enzyme. So here you can see some specific examples. So first, high levels of NADH inhibit citrate synthase, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, as well as the pyruvate dehydrogenase complex. This is consistent with the cell has high levels of energy, so we've already made a lot of NADH, but that all of that NADH hasn't yet been converted to ATP. So it suggests that the TCA cycle is backing up. So this is going to inhibit these enzymes. So in addition, sensors that the cell is low on energy can also activate these enzymes. So for example, ADP can activate isocitrate dehydrogenase, which suggests that we need to feed more intermediates through the TCA cycle in order to make more energy. Calcium also activates alpha-ketoglutarate dehydrogenase, isocitrate dehydrogenase, and pyruvate dehydrogenase. This is also a sensor that the cell needs energy because many times release of calcium is required for different cell types to do work. For example, release of calcium, it triggers muscle contraction, so that suggests that the muscles are going to be highly active and are going to need a lot of ATP. So this can also serve to activate these enzymes. One more inactivator, like NADH, ATP tends to inactivate alpha-ketoglutarate dehydrogenase, isocitrate dehydrogenase, citrate synthase, and all three of these enzymes are inactivated because this is a sensor that the cell is high on energy. So in all of these cases, the enzymes are being inactivated if the cell has enough energy in the form of NADH or ATP, or sometimes by downstream products as well. And these enzymes tend to become more active if the cell needs energy, which is indicated by calcium, indicating that those are actively working cells, or by high levels of ADP, indicating that the cell is low on ATP. So as I mentioned, it's important that these enzymes are highly regulated because multiple pathways actually integrate through the TCA cycle. So for example, oxaloacetate, we've already talked about that this pathway can be used for gluconeogenesis. In addition, other pathways can feed in to make oxaloacetate, so lots of different amino acids are broken down into oxaloacetate. And different amino acids can also feed in as fumarate, succinyl-CoA, alpha-ketoglutarate. So amino acid breakdown can feed into several different places. In addition, fatty acid breakdown can be used to make succinyl-CoA, so we can feed in energy from proteins as well as fats into the TCA cycle. And TCA cycle intermediates can be used, as we mentioned, for gluconeogenesis. They can also be used to make fatty acids and can be used to make cholesterol. They can also be used to make some amino acids, as well as the porphyrins that are used to make heme groups for hemoglobin or for other enzymes such as those that are part of the hemes that belong to the electron transport chain. So regulation of the TCA cycle is really important because it can help to determine whether energy should be sent, whether we have enough energy and should send these intermediates towards the biosynthetic pathways shown in red, or towards the energy making pathways, so the catabolic pathways shown in green. So that's going to be the end of this video. So thank you for paying attention. Don't forget to bring your extra credit bonus point in on Friday. Also, don't forget to bring your notes that you're going to be using to go through and trace those carbons on the TCA cycle. So also, finally, please bring any questions you have about these videos and I'll be happy to answer them in person in class. Thank you.